My name is Isabel Dominguez, and I am a researcher at Boston University School of Medicine. I am delighted to be here with all of you today, and more because Ms. Sills, the president of the CSNK2A1 Foundation, asked me to prepare this short overview of CK2, its history, its discovery, and future research. First, I would like to introduce two terms that probably are already familiar to you, that are CSNK2A1 and CK2 alpha. CSNK2A1 is the name of the gene that is sort of the blueprint to make CK2, that is a protein. The CK2 protein, the CK2 alpha protein, is the molecule that works in our bodies. And this is the protein that we are going to be talking about in the next few slides. CK2 alpha was discovered around the late 60s. And in many different kinds of scientists have been studying it since then. And that includes geneticists, biochemists, structural biologists, cellular biologists, developmental biologists, chemists, and others. And in the next two slides, I'm going to summarize what all of these researchers have done since CK2 was discovered. First, what we know already is how large CK2 is. It contains 391 building blocks that we researchers call residues. They are this. But CK2 doesn't look like this, where the building blocks are one after another. It actually has a three-dimensional structure, and it actually looks like this. For simplification in my talk, I may have represented in some slides like this, like an oval. What we know also is that CK2 is always working. It is like the refrigerator in your house. It's always working. It's not like the light switch that when you switch on your lights, it really is like the refrigerator. We call this being constitutively active. This makes CK2 alpha a very special protein. Most of the proteins are not working, and then they only get activated when needed. CK2 in that regard is very special. CK2 is also special because it can work alone on its own. Is what we call a monomer, from mono, one, mer, unit, or it can also work together with CK2 beta. That is a protein that regulates its activity, and forming what we call a tetramer, so tetra, 4, mer units, so 4 units. This is how these 4 units look like. So one alpha here, one alpha there, a beta here, and a beta there. This is the scheme that I may have used also in the presentation to represent this. And to this complex, we call CK2. So we have CK2, that is the complex, CK2 alpha, and CK2 beta. You may be asking what CK2 does. So what we know is that CK2 protein acts on other proteins and changes them in a way that it either activates it or then deactivates them. And once these other proteins are activated or deactivated, these proteins do something in our body. So basically it helps our body respond to things. And we will go into this in a few more minutes. And you may be asking where CK2 is. And the answer is everywhere. It's everywhere in your body, and it's everywhere in nature. And I have shown pictures here of yeast and plants, flies, mice, frogs, and fish. And all of these many different organisms contain CK2, and it works very similarly in all of these organisms. And we scientists used all of these organisms and many other ones as tools to study CK2 in all of these very different contexts. For example, in my laboratory, We have used these tools that we call in the laboratory models. We have used mice and frogs to show that CK2 alpha is very important during the development of the embryo. And you can see here an embryo that has a full amount of CK2, and you have here an embryo that is lacking CK2 alpha. And you can see that this embryo is a little bit different from this other one. We have used similarly frog embryos for the same purpose. And you can see here that this embryo here that has less amount of CK2 alpha is different from this one that has a normal amount of CK2 alpha. We have also used cells that we can have in our laboratories, and we can put them in plates and culture them, cells that come from our bodies or the bodies of animals. And we have figured out that CK2 helps receive signals that come to the cells. And signals we call a number of molecules like hormones, for example, like insulin. So those are signals that come to the cell and tell the cell, you need to do that now. So CK2 helps interpret those signals and have the cell respond to those signals in the particular way that the signal asks the cell to do. Recently, in 2016, Drs. Okur and Chang discovered that there were patients that have problems with CK2. And what they have is what we call variants, so CSNK2A1 variants, that all it is a change in the blueprint that makes CK2 alpha protein. And if the blueprint is changed, then the protein will be changed. And what happens in some of these residues, some of the building blocks that I told you about before, are mutated. And all that means in the scientific jargon is that they are changed. And this new protein works differently from the one that doesn't contain those changes in the blueprint. What we are trying to do now is we are trying to figure out how these new proteins that have these changes work. And the strategy that we researchers follow for OCNDS and for any other disease is relying on the patient's information. So we take from the patients and their families genetic and clinical information that we can use to work out how these changes change how the protein works in these bodies, in these animals or in these cells. We can also take patient cells and tissues and explore them and explore how the cells respond now that this protein, in this case CK2 alpha, is changed. And then with the results from the cells from the patients and the results from the animal models in the laboratory, we will have discovered the mechanism of how those changes make a difference in the patient. And chemists and other engineers and so on and so forth, a number of other scientists will be designing therapies that we will bring back to the laboratory and then using them in the patient cells or in the animal models to see if they work, if they can make the changed protein work better. And once we have seen that it works better both in the patient cells and the animals, then we can use those as therapies that we can give to the patient, either to prevent or to alleviate or treat the symptoms that they have. For example, one of the things that we are doing in my laboratory is we are injecting the mutant proteins, the proteins that are changed, the CK2 alpha proteins that are changed, in the very early embryos from the frogs. And here you can see an embryo that has 8 cells. Here you can count them, 4 on the top and 4 on the bottom. And then once we inject this protein, it will have an effect later on when the embryo is already a tadpole. And then what we can do is we can see what changes has happened due to this protein, this new CK2 alpha being present in this embryo. And then not only we can see what morphological changes have happened, but also we can investigate how these changes occur. And once we know how these changes occur, and that's what we call the mechanism, we can see how we can, again, design therapies to treat these symptoms. I just wanted to summarize with this last slide what I presented to you today. I presented to you some information about CK2, how much we know about CK2 alpha, and some about CK2 beta, and how the two of them form this beautiful tetramer that is represented in here. I have talked to you about some of the tools, what we call the models that we use in the laboratory that are available to us, from the mice to the yeast, every single one of them, that allows us to discover things about CK2. And we use to investigate the changes that are happening in CK2 alpha in patients that have OCNDS. And hopefully with our investigations will lead to a discovery of the mechanism of how these changes act in patients. For that, as I said earlier, we need patient and family contribution. We need your information so that we are not investigating blind or with our eyes closed, but we have all of the information, the largest number of information that we can get, so that we can appropriately do the experiments. And we do all of this in collaboration with the CSNK2A1 Foundation. That is the bridge between the patients and ourselves, that it funds our research and also encourage us to continue working for you. And this is the end of my presentation for our meeting. And I'm very happy to take any questions during our Q&A later. Thank you so much for listening to me.
Good afternoon, everybody. So I'm Heike Rebholz. I'm working in Paris at the Institut de Psychiatry and Neuroscience, so Paris, France. I have been working on CK2 for quite many years. And first I want to thank the Simons Foundation and the CSNK2A1 Foundation for giving me the opportunity to talk, present my work on CK2, and tell you what we're doing in particular on OCNDS. But let me start with giving you a brief background on CK2. CK2, as you probably know, is a protein, and the protein is a three-dimensional structure that is composed of amino acids that are attached to each other to form a chain, and then to form a three-dimensional structure. CK2 is not only a protein, but also an enzyme. And what an enzyme is, it means it's a protein that has a job to do in the cell. It has a function to fulfill, an activity to perform. And the activity that CK2 is supposed to do is to be a kinase, which means transferring a phosphate group from one substrate from one phosphate donor to a substrate or acceptor protein. And this is important in the cell since phosphorylation can be occurring very quickly and can be like a light switch to turn on or off activity. And it can also happen that to turn off that activity, dephosphorylation occurs similarly in a very fast manner. So phosphorylation is a means of a cell to control various proteins in their activity upon demand in a very fast and regulatable manner. Okay, so CK2 is therefore a kinase that can regulate other protein through the process of phosphorylation. It is also important to know that CK2 is expressed in every single cell of our body, and is not only a protein composed of a single amino acid chain, but actually it has 4 amino acid chains that are all forming their own three-dimensional structure, and that are then forming together a heterotetramer. So 4 units, 2 of which being a catalytic subunits, together form the holoenzyme, the full CK2 enzyme. So when we study CK2, we usually look at the whole enzyme, not only just one chain of amino acids, but 4 together. And CK2 is expressed, as I said, everywhere, but in the brain, we know it's highly expressed, and there's a lot of CK2 activity in the brain. CK2 regulates many, many functions in our bodies, in every cell. We can start, for example, with cell growth and division, with metabolism, with cell-to-cell communication, cell shape and structure determination. And it's important in cancer, and it has been really well studied in the last decade or so in cancers. And for example, inhibiting CK2 is now a novel approach to target or to treat certain cancers. They are responsive to CK2 inhibitors and shrink in their growth. And even in COVID-19, CK2 has been shown to be involved, meaning that it helps or is part of the mechanism by which the virus enters the cells and amplifies in our own cells. So CK2 is encoded by the gene called CSNK2A1. As you might know, every protein is really encoded in every cell in form of the blueprint, the DNA. And it sometimes happens that this DNA can accumulate mutations, little changes, little faults in the sequence of the DNA. As you can see here, there's a correct copy. But then when, for example, the DNA is amplified, when a cell is dividing, for example, then it can happen that a mutant copy, a mutant is entering in the DNA chain. Normally, it is recognized by an apparatus for repair, but sometimes this apparatus of repair can miss certain mutations, and so these mutations remain permanent. And when the mutations occur, these mistakes occur early on in the development of an organism, then of course these mutations are present in all cells of the organism that is developed, and therefore that will be manifested by disease or pathological symptoms. So on a very basic biological level, you can imagine that when a protein composed of correct amino acids, when it has a different amino acid, which will take up a different space and have different electrophysical properties, it will also change the three-dimensional structure of that protein. It could alter the structure as to open up, clefts as to introduce kinks, as to make the structure more compact, etc. And all of this will affect how that mutated protein will interact with other proteins in the cell and how it can exert its function. So, as I told you, CK2 is expressed everywhere in our bodies, but it's highly expressed in the brain. And here I'm showing you just some slices from our mouse model, from the model that we're studying in the lab, the mouse. We stained these mice with antibodies recognizing different subunits of this CK2 holoenzyme, and you can see that basically every structure in the brain, not only mouse but human, they have CK2. And many proteins that are phosphorylated by CK2 in the brain are very important for functioning of the brain in the healthy state, but also in the diseased state. So here I'm showing you mutations as they have been described to occur in patients with OCNDS. This is from a paper from 2018, so it might be not entirely up to date, but the major mutations that have been found in patients are noted here, and you might appreciate that they're clustering mainly here in the front, in the beginning of the gene or in the first half, which is very important for the functioning of CK2. So that means that you can imagine that if amino acids that are in this important sequence here are occurring, that they might also impact on the function of CK2, on this kinase activity. And this is what we actually started out doing. We made this protein CK2, either the normal protein or the ones that are containing one of 16 different described mutations. We purified these proteins either from mammalian cells or from bacteria, and then we tested in the dish, in the lab, in vitro it is called, the activity of CK2 towards a substrate. And we could find that all of the mutants described to be linked with OCNDS have a reduced activity, really all of them, even when they are co-expressing, when they're the holoenzyme, but also when we only use one subunit of these different chains that make up CK2, which is the catalytic subunit. So in both variations, either CK2 alpha alone or as the complete enzyme, we find that the mutations lead to reduced activity of the kinase, which is not all too surprising given that we already know where these mutations occur in the gene of CK2. Here you see the localization of some, but not all of the mutants. Mainly, I want to show you that, for example, here the normal CK2 has a very spot-like presence in a mammalian cell, and so do most other mutants, but some of the mutant proteins, you see it here in a better, bigger version, they do not at all look within the cell like the wild-type protein. So not only do they not really express in the same location, but there seems also to be less protein present, which means they're less stable. So here you can see on that plot here, on that graph, you can see that the localization of these two mutants has changed from being what we call nuclear in the very center of the cell to being more in the what we call cytoplasm of the cell, so outside of the center. We also studied, we generated and studied over the many years, we studied different mouse models in which the kinase CK2 is lacking or is knocked out, so to speak, in certain brain cells. So these are different knockouts that we generated where CK2 is lacking in this kind of cell type, in the cells that express DRD1A, you don't need to really know the details, or another line and yet another line. Certain cells in the brain lack CK2. And for example, with this mouse line here, we do find that they have we call it phenotype, that they have behaviors that are reminiscent of OCNDS symptoms. For example, they're hyperactive when they are exposed to a novel environment. They exhibit stereotypies, which are uncontrolled movements, mainly of the limbs. They have a change in circadian rhythm and a decreased memory. They also have motor deficits and motor learning deficits. So currently, we are actually studying the first OCNDS mouse model. So we chose to introduce the most commonly occurring mutation in OCNDS, which is this one, K198R, in mice. And we studied these mice for behaviors that are phenocopying, that are similar to what we see in patients, just like what I showed you for the other mice. Except now we're really working with mice that are genetically very similar to the OCNDS patients. And we find here that, again, spatial memory in this test, where the mouse has to choose between different arms of a Y maze and has to remember where it has been and explore the other basically arms, they show the OCNDS mice, they show reduced capacity to memorize the localization. They're also in an open field environment, they're less active in the first 20 minutes, which could indicate that they are maybe less anxious. And they have another difference when we put them in an open field, in an environment, in a box basically, and we introduce a novel object. The mouse is very curious. The OCNDS mouse explores that novel object more than the normal mouse. It also is less anxious when we give it the choice of spending time in that bright lit area of a box or in a dark area. The mouse, as you know, they live at night, and so they're nocturnal. They prefer normally to stay in the dark room. And there's a slight difference between the wild-type mice, the normal mice, and the OCNDS mice. We're studying also how neurons develop, how they look when they're derived, when they're coming from the mutant mice, so the OCNDS mice versus the wild-type mice, and we can see that they have changes. The neurons, they have long protrusions that are branching up at the end, and we can see a trend. We're studying that more to define more, a trend of a differentiation effect in the mutant mice, or in the neurons in the brain cells that are derived from the mutant mice. We're going to do, in the very next step that we're doing, will be to look at these neurons really in the brain itself by performing three-dimensional microscopy on fluorescently on labeled cells, so that we can see in the context of the brain if there are changes in how the neurons develop, how they integrate into a cellular context in the OCNDS model mice. So in summary, CK2 activity is reduced with all OCNDS-linked mutations. Sometimes these mutations lead to abnormal localization in cells or a reduced stability. And we're currently assessing the symptoms, the behaviors of the OCNDS model mice in order to also be ready to next explore where the therapeutic approaches are successful, because now we have a mouse model that we can use to study and test new therapies, new drugs, maybe gene therapy. So this will be the next work that we will perform in the lab. I want to thank people in my lab, especially Jose Cruz Gamero, who's working very hard as a student of mine. And I would like to thank all my collaborators, some of which are talking also at this meeting. And I want to particularly thank Jennifer Sills from the CSNK2A1 Foundation for her continued support, mental, and also for funding, of course, our work. So, and thank you for your attention, and I'm ready for questions later.
All right, good afternoon, everybody. I hope you had very informative sessions so far, which looked to be the case to me when I look at the agenda. And now I'm gonna talk about the potential clinical studies that we have in our mind and actually have started to reach out to you. Before going deep into our first study, I would like to mention a little bit about the basics of genetic syndromes. So a syndrome is a set of concurrent things that usually form an identifiable pattern. In here, like the pattern is the symptoms or findings that your children have experienced. And I would like to divide the genetic syndrome, so because a syndrome can be in any other specialty, into four major aspects. The first one is the cause. So what is the underlying cause? So in our terms, like what is the gene that is causing the syndrome? And what are the variants? Is there any one specific variant, or is there a set of variants, different variants that we should test for? And in our case, like from the get-go, we knew that the variants in CSNK2A1 gene are the responsible cause of the problems that the first initial 5 kids experience. And then we have distinctive features, so which help us even before ordering a genetic test to narrow down our differential diagnosis, which I will talk about in a little bit more detail in the next slides. And the third aspect is the natural history, which is what other problems might develop in these individuals and at what frequency, once a week, once a month, or like just once, twice in a lifetime, and when, for example, if there is a seizure or regression, when they occur, like what in terms of age. And hopefully the Simon's VIP study will answer many of those questions in terms of natural history. And all of those have like one main goal, which is the management, the ideal management that would help those individuals to reach their potential. And ultimately, if there's any medication, like drugs, or more recently, gene therapies that could stop or reverse the health problems that our kids are experiencing. So what I mean by distinctive features is, so far we know that there are a bunch of problems that kids with Okur-Chang neurodevelopmental syndrome experience, but from the global genetic disorders perspective, they do not specific to Okur-Chang. So what I mean by that is that we do not have a distinguishing feature, such as I show here, that these ear creases or like increased hair on the forearm and elbow of some kids who also happen to have intellectual disabilities and developmental problems. So as clinical geneticists, when we see these type of small features, we can narrow down to our differential diagnosis to a few specific disorders, genetic syndromes. And this could be sometimes also specific movement patterns, like, for example, arm flapping or tics, or like the certain age when, for example, regression starts, etc. So as a result of this non-specific nature, when we search sort of our yellow pages for genetic syndromes by the two most common problems in these kids, the speech delay and intellectual disability, we get over 1,000 entries, which is different syndromes. So, and you can imagine that narrowing down your differential diagnosis from here is a really challenging task. And what we used to use historically is the recognizable facial appearance of individuals, which in our field called facial gestalt. Gestalt is a German term for shape or form, and the dictionary definition is an organized whole that is perceived as more than the sum of its parts. For example, here I show a girl with Down syndrome, a disorder, the most common genetic disorder that could be recognizable by the entire world. When we first see a photograph of a kid with Down syndrome, for example, we first think about Down syndrome. And then when we look further, we think about we define the eye, the shape of the eye, or eyebrows, the cheek, nose or mouth, etc. But we do not go into this at first. So this is what facial gestalt means to us, what we think of an individual, like in terms of their diagnosis, potential diagnosis, when we first see their face. So starting with our initial publication, although at the time we were not aware of any common facial gestalt, that we were given permission to publish two pictures of one girl at two different ages. The left one is at 3 months old, and the right one is around 3 years old. We thought that this would help for featured definitions of the facial features. And for example, recently, there is another study from Spain by these researchers. They also published the pictures of their patient at two different time points. And when you look at, or when we look at these two kids, for example, when they are at similar age, they seem to have like a similar facial appearance. And also when they are both toddlers, that we can appreciate the similarities. And of course, like there are differences in between in other publications, that not every kid has this type of facial appearance, but we also tend to account for familial and like ethnic factors as well. But from both personal experiences with my interactions with you families and looking at some other papers, I can say that we are able to, I am able to see this emerging facial gestalt that will help me to recognize the syndrome. So for this reason, we want to test this hypothesis, which we think there is something, that if there is a recognizable facial appearance among the kids with Okur developmental syndrome, and if there is, which we believe there is, how this facial appearance changes over time, which means like from infancy to adulthood, as the physicians would see individuals at different age points at different times to reach the diagnosis. And this would help us. And like I said, although now with the new sophisticated next generation technologies, the focus, the diagnostic method kind of shifted. Like first gene comes rather than the syndrome definition like in before, but still the face would be helpful, especially when there isn't a widespread access to those next generation sequencing technologies like whole exome or whole genome sequencing. So this will help us to educate other geneticists, and when they see another individual with similar symptoms, by looking at the pictures and comparing them with their patient's face, that they will narrow down their differential diagnosis. And for this, like, there are some online tools that would help us. It's not easy nowadays that among the 4,000 different genetic syndromes that to remember everything. And like for those tools, work in a way that you enter the findings of your patient, and optionally you can also add some photos if the family consents, and you can get a differential diagnosis sorted by the highest possibility from the lowest. And you can see here that when I entered these, the Okur-Chang comes at top. But there's a reason for this, for example, the decreased circulating IgA level. So this was the finding that we reported in our first paper. But with time, since the last 5 years, although some of our kids are experiencing immune problems, this finding is not that much common, maybe not at all. So if we remove this feature from this set, the Okur-Chang's order might go to back, and one might really overlook. And if there is a facial representation here, like for these three syndromes, again, by looking at those, sorting through those differential diagnoses and looking at those pictures, another clinician might come to a conclusion, or at least a differential diagnosis, a very strong differential diagnosis for Okur-Chang syndrome, which would help us to increase our family and to study more features about this syndrome. And secondly, which actually we are doing these days on the back end, like whenever a new family or a physician reach out to us and ask about a novel variant which has not been reported so far and which does not seem to be in a very important region, we want to see the kid's face, so to see that whether she or he looks like other kids in a way. So, and this would help us. Of course, it's not gonna definitely lead to diagnosis, but this would help us to say more confidently that yes, Okur-Chang might be the most plausible answer along with this genetic diagnosis. And for our second study, we would like to look at the different variants other than the ones that we most commonly see nowadays and the rest of the field are aware of. So just to recap and a reminder of those variant types when we talk about them, those technical terms. Here, I got this table from this website. I think it's a really nice representation. That the normal gene, which in the scientific jargon wild type, has this sequence, like one big fly had one red eye, which is like a good structured sentence. And this is a good coincidence that all the words are composed of 3 letters, like our amino acids, like here R, K, etc. And the machinery in our cells, they also read those amino acids codes in this 3-letter format. So when we talk about the missense variants, this means that one of these letters in one of these amino acids and words here, that they change to another one. So, and then this word loses its meaning. And in real life example, the most 2 common variants here, they are missense variants. And when we talk about a nonsense variant, there is a premature dot here that results in the abruption of the sentence, and we only have the one big. So for this cell, this doesn't mean anything. And it disregards this signal and behaves like as if there is no signal. And in real life examples, for example, test results like this, the X at the end, which stands for termination, and sometimes in the 3-letter form, you can see ter as well. These are nonsense variants. And for frameshift variants, either 1 or 2 letters might be inserted, added here, or deleted. And in that case, the machinery is still it shifts the reading frame to the right, and you will see that the meaning of the remaining words here are lost. These 2 are preserved. but these will still behave like this top one nonsense. And like you will see in real life examples in your test results, like this type of results. And then we have deletion variants. This deletion variants might involve only one word, one amino acid, in this case here, fly, and in this real life examples is like this D here, or it could involve a part of the gene, which you can see in your reports like exons 2 for deletion, or it could encompass the entire gene along with other neighboring genes. And in your test results, you can see that like this. And in these results, you may also see sometimes if the deletion is partial in the explanation, text explanation, that this deletion affects exons 2, 4, 3, 5, 6, etc. And as you know, and recently Dr. Dominguez and Dr. Rebholz reported in their functional studies paper that the missense variants are the majority of the variants so far. And we get a lot of queries about other variants whenever a family or a physician encounters, finds out that their kid or patient has one of those other variants. So for this study, we want to enroll kids with this type of deletions, mainly the red box, and also these orange box variants, nonsense and frameshift. They behave like this deletion variant. So we can also enroll those individuals as well. And we want to study both, like we want to catalog the variants, and then we want to see what are the health problems. And by this, we believe that we will increase the awareness and knowledge among the wider community, both families and other physicians. And we will study the differences in the findings and symptoms, whether that makes there is any difference between missense and non-missense variants. And this would also help us to understand the further cellular and biological mechanisms. For example, for those missense variants, again, from Dr. Dominguez and Rebholz study, we know that there's some sort of loss of function. So these other variants, they are also called loss of function, but do they show their effects in the same way, etc. And again, in turn, this will also guide our treatment strategies going forward. So with this, I would like to stop here and wrap up. And I hope to see you in our live discussion, and I hope to answer your questions. Thank you.